Hi everyone, and welcome to this session where we're going to look at biosynthesis of non-essential amino acid in the series of protein metabolism. By the end of this session, we expected to be able to describe synthesis of non-essential amino acid and also disorders related to metabolism of amino acids. Non-essential amino acid can be synthesized from intermediates of metabolism or from the essential amino acid. And we start off with the synthesis from alpha keto acids. And here we see that amino acids are synthesized by transfer of an amino group to the alpha keto acid. Pyruvate is an alpha keto acid, and when it takes up an amino group, it's converted to alanine. We also have oxaloacetate as an example of an alpha keto acid. When it takes up an amino group donated by glutamate, it forms aspartate. Alpha ketoglutarate, it takes up ammonia. In itself, it's converted to glutamate, and this reaction is catalyzed by glutamate dehydrogenase and requires NADPH or NADH as a cofactor. Also important to note that glutamate can also be synthesized by the reverse oxidation of deamination, which is catalyzed by glutamate dehydrogenase when the levels of ammonia are high. We have other proteins or amino acids which are synthesized by amidation. For instance, we have glutamine, which is formed from glutamate by glutamine synthetase. Here we see glutamate picks up ammonia and is converted to glutamine. And this reaction is catalyzed by glutamine synthetase, requiring ATP as the source of energy. Asparagine is also formed from aspartate, and we have glutamine as the amide donor. So glutamine donates the amide group to aspartate, which is then converted to asparagine. This also requires ATP, and the reaction is catalyzed by asparagine synthetase. We also have some amino acids which are synthesized from the intermediates of glycolysis. We have serine. Serine, through various reactions, that is through the 3-phosphoglycerate, is converted to serine. And serine can also be synthesized from glycine through a transfer of hydroxymidane group by serine hydroxymidane transferase using methylene tetrahydrofolate as a 1-carbon donor. In the same reaction, we can form glycine. So glycine can also be synthesized from serine by removal of a hydroxymidane group by serine hydroxymidane transferase and tetrahydrofolate is the 1-carbon acceptor. So it's the reverse. When you're forming serine, the tetrahydrofolate is a 1-carbon donor, whereas when you're forming glycine, tetrahydrofolate is a 1-carbon acceptor. We also have formation of cysteine, which is synthesized by 2 consecutive reactions in which homocysteine combines with serine, forming cystathionine, which in turn is hydroxide to alpha ketobutyrate, which can then be converted to isocyanate-CoA and cysteine. It's important to know that methionine is an essential amino acid. And so, since homocysteine is converted from methionine, cysteine synthesis can only be sustained if the dietary intake of methionine is adequate. We look at proline. It's synthesized from glutamate and through various reactions, which also involved spontaneous cyclization, we form proline. Tyrosine is also formed from phenylalanine, an amino acid, and through the activities of phenylalanine hydroxylase, phenylalanine is converted to tyrosine. And this reaction requires a molecular oxygen and also tetrahydrobiopterin. which is synthesized from GTP. So let us look at the metabolic defects associated with amino metabolism, and these are inborn errors, which are caused by mutagens that may result to abnormal protein, and here we're talking about the enzymes in particular. So the inherited defects may be expressed as a total loss of the enzyme activity or a partial deficiency in the catalytic activity. So newborns can be screened for a number of treatable disorders, and this is done by obtaining blood from a heel prick, and then tandem mass spectrometry is then performed. So we start with phenylketonuria, also known as PKU, and this is as a result of defective or deficiency of phenylalanine hydroxylase, which is involved in the formation of tyrosine. And this are defective in this enzyme will result to accumulation of phenylalanine. So major clinical features include mental retardation, that is, if it's untreated, you have seizure. You also have hypopigmentation. Remember, tyrosine is involved in the formation of melanin. So for treatment, you use low phenylalanine diet and also avoid aspartame. We have also alkaptonuria, which is as a result of a defective enzyme that is an oxidase, and this will result to accumulation of the homogenetic acid, bringing about dark pigmentation, therefore turning urine urine turns black on standing. It will also result to degenerative joint diseases. Treatments: low tyrosine, low phenylalanine diet, also vitamin C may slow pigment formation. There are other disorders associated with the formation or the breakdown of phenylalanine to acetoacetyl-CoA, and these include the tyrosinemia 1 as a result of the defect activity or deficiency of tyrosine aminotransferases. We also have tyrosinemia 3 and also tyrosinemia 1. So these are disorders associated with deficiency of this enzyme in the breaking down of phenylalanine to acetoacetyl-CoA. We also have another set of disorder known as the maple syrup urine disorder, and this is a result of deficiency in the branched chain alpha ketoacid dehydrogenase complex. And this enzyme involved in oxidative decarboxylation of alpha ketoacid from leucine, isoleucine, and valine, which are converted to acyl-CoA derivatives. And this will result to accumulation of branch chain amino acid and also keto acids, which are neurotoxic. And various clinical features are manifested. These include the maple syrup odor of urine, poor feeding, vomiting, and also seizures. Treatment include restrictions of branch chains amino acid in diet. Thiamine supplementation may also help since they are cofactor for this enzyme. We have albinism, and this is as a result of deficiency of tyrosinase, which is involved in the breakdown of tyrosine. Remember that tyrosine is involved in the formation of melanin through the dopa-dopaquinone pathway, and therefore the melanin biosynthesis in the melanocyte is defective. And this may lead to failure of melanin formation, resulting to hypopigmentation. And this is followed by clinical features such as pale skin, white hair, light eyes, and also photophobia. We have the cetonuria, and this is as a result of defective renal tubular and intestinal transport of cysteine, ondine, lysine, and arginine. And these transporters, or the renal tubula, they assist in the reabsorption of these amino acids. And so we have defective or poor reabsorption and leading to excess cysteine in urine, which may crystallize to form stones. And the clinical features associated with these disorders include the recurrent kidney stones and also the hexagonal cysteine crystals in urine microscopy. Treatment for this disorder include hydration and also urine alkalinization. Homocystinuria is as a result of a defect or deficiency of cystathionine beta synthase, which is a vitamin B6 dependent enzyme. Remember, it's normal reaction, or it catalyzes the condensation of homocysteine and serine to give us cystathionine, which leads to the formation of cysteine. And this disorder will result to accumulation of homocysteine in the vascular and also connective, resulting to vascular and connective tissue damage. Major clinical features include lens dislocation, skeletal deformities, and also mental retardation. And so with that, we come to the end of this session, where we have looked at various biosynthesis pathways of non-essential amino acid and also disorders related to amino acid metabolism. Thank you.